All right, so the pre-lab, it gives an experiment. It's very similar to the one that you're going to do. And it's a reminder, a review of the terminology. What is a control variable? What is an independent variable? What is a dependent variable? So the dependent variable, the way I remember dependent variable is the dependent variable is what you write down on paper. Like it is what you are monitoring. It's the data you're collecting. So if we want to see how temperature changes, and then in our data table, we are writing down temperatures, that is the dependent variable. Like what are you writing down? And then the independent variable would be whatever is having an effect on the dependent variable. I think that's all I want to say. So I give you an example. I don't want to say what the answers are, but you are looking at this example lab. It's similar to ours, but it's not the same thing. And then you will say what the control variables are and what the independent and dependent variables is or are, okay? And if you don't get it right, then you get to try again. You're meant to learn something.
Are you in class?
Yes. Hey, Mary. Oh, okay. Sorry, my daughter and one of my former students is here. Okay. They're learning about titles. Titles have to be descriptive, right?
Titles have to be very descriptive.
I'm not lying.
No, it's nice because the more descriptive your title is, the less writing you have to do in your lab report.
Yep.
So it's actually saves you time.
Mm-hmm.
Have they met the dogs, your class?
No.
Your class hasn't met our dogs?
No, I'll have to do that later. This has been a really long lecture. I feel really bad. Okay. All right, now then what you see here is what you need to make sure that you do when you make a graph. So bye, y'all have fun. Your graphs actually will be a big part of your data analysis grades. So because graphs are incredibly important. So make sure that when I ask for you to graph data, that you have everything that I've written here in Model 3. You will need to have a very, very good title, label the axes, have the units, appropriate scale, a legend, and then you're going to have to make some decisions on the graph itself. Sometimes you need to connect the dots, okay? Sometimes it's appropriate to connect the dots. Sometimes it's appropriate to make a best fit line. So you're gonna have to figure out, is this the type of graph where you would connect the dots, or is this the type of graph that you would do a best fit line? Some data, you include the origin as a data point, but a lot of experiments, you don't use the origin as a data point. So you'll have to think about that. So we'll be summarizing a lot of different types of data. So these are the kind of questions you'll be asking yourself throughout these 10 weeks. Okay. And then I have some just more questions about general experimental design. And then here is another description of how to write a title for a graph. And then you get some practice writing some titles. Then you get practice writing some results, and then some general questions about data analysis. On the ACT, for sure, the science section is all data analysis. It's all data analysis. You really don't have to know any science, but you have to do data analysis. So I've given some sample questions, and then I just tell you the kind of lab we're going to do. So this is a variation of Texas A&M's general chemistry course, and it is a review of experimental design. What's the independent variable? What's the dependent variable? How do you collect data? That kind of thing. So if we go over to the actual lab itself, here are the steps and instructions for the lab itself. You will notice, because this is a college, this is a freshman college chemistry lab. We're doing part of it, not the whole thing. And so you'll notice some terms, etc., that you have just probably seen very recently on stoichiometry and balancing equations. And so that was on purpose that you learned that, because I have worked as a research scientist, and you do have to balance equations if you are a chemist and you're synthesizing a molecule. You actually really have to do this stuff. And I'm not going to test you on that, but that is part of the lab. If you would have done this in college, I go ahead and give you all the information that you need. So I left this in here just to show you that it's not just to torture you, that's actually really what scientists do. And then you'll notice the list of materials here are found in your laboratory kits. So you have a 25 ml beaker, and then you have, I think I also give you all a 10 milliliter beaker as well. They make 5 milliliter beakers. It's so cute. Okay. And most of y'all have a balance. A few of you don't have a balance, and that's okay. I'm going to get you, I've ordered more, and they should be here, and I'll hopefully get them to you this week or next week. You don't really need a balance for the first lab. And then you should have a small bottle of vinegar. And then you should have a container that says Alka-Seltzer, and you've got an Alka-Seltzer tablet in your container. And then you should have a little bag with 3 different types of scoopers, okay? I couldn't decide which one I wanted, so I bought all 3, and then we're gonna decide which ones work best. And we will also use them to stir. And then you should have some weighing paper as well. We're going to learn how to use weighing paper. So I would like for you to take a couple minutes, go get your lab supplies, and then come back. And then I'm going to show you how to use everything. And then I will start the lab. So if you want to do the lab now, you can, or you can do it later. But I do want us to go through all the materials. So go get your supplies and then come back, because there's actually a certain way to use weighing paper that I will show you. So, okay, take 5 minutes, go get your stuff, and we'll meet back here. Oh, yes, good question. You do not have anything toxic, but some of these can stain. We're going to use food coloring and stuff. So yeah, you can do everything at your desk. You just need a space that you can work. And then if it is something like a desk that is really, really nice, you may want to put down either like wax paper or paper towels, can put down. So go ahead and make yourself a little space. Your stuff's out here. Yes. Okay. So, yeah.
Did you lose it?
How did I? Is there like a box somewhere? I had yours. See, mine. Did you take this in your room?
No.
Oh, here it is.
Oh, look at that box that was clearly away. Reaches.
Make one work here.
Is it the third?
Yeah. I wish I could see everybody's, and I don't know how to get that to go.
When you click on it, there should be like an extension you can download.
Oh, is that it? Is it that? Oops. This?
Yeah.
Chris Gamble's official release?
Yeah, I'm pretty sure that's the fix.
Oh, okay. I'll do that later. Let me see like a couple figure. Let's see. Okay, no, we got water. I know I love this. This one is really fun. All right. When you're ready, so everybody I'm sure knows I play video games and I've played World of Warcraft. And when we're ready for our, to fight the boss, we put an R in the chat. What? So when you're ready, if you could just put an R into the chat, then I'll know when to begin. Thank you. Diego's making fun.
Well, do you know what R is short for?
Ready.
Oh my God, I don't believe it.
Is that used in other games?
No.
Oh, see? Yeah, I'll learn something. No, I was saying BRB before you were born. Okay, 1, 2, 3, 4, 10, 11, 12, 13, 14. We can make a guild.
No, we don't.
All right. So in your lab kit, you'll need, actually, I'm gonna stop presenting. I can see. Now I can see everybody. Okay. You need your water. So find your water bottle. And find your vinegar. The graduated cylinder. Your 25 ml beaker. Your Alka-Seltzer container. Your balance. What did I do with those? Did I give you the stirrer things? What did I do? I should not be allowed to hold anything in my hand. What did I do with them? Luna, did you eat them? Where are yours? How did I lose them? I seriously just had them a second. Okay. Okay, Now, you have other things in your lab kit. One of them says water lab, and this is for the water lab that we're gonna do next week. So you don't need this right now. You have another little bottle that says magnesium hydroxide. That is actually milk of magnesia. So this is an over-the-counter, and when you get old like me and you get acid reflux, you drink this. So this is not dangerous, but it is magnesium hydroxide. We don't need this today. We'll need this next week. I think, and then get out the, you should have a little bag that says weighing paper. So go ahead and get out a piece of weighing paper. I bet I'm sitting on it. Okay, this is weighing paper. And the way you use weighing paper is you fold it and make a triangle like that. And then you open it back up. So now it has that crease in it. And then this way, when you weigh out your sample, when you pick it up to put it in your beaker, it doesn't fly off on the sides. It's just easier to put it into your beaker. So in general, whenever you use weighing paper, always fold it in half like this. So you get that crease. And then it just makes putting the samples into the beakers very, very, very easy. Now, the first thing we want to do, can you hand me my mortar and pestle up there? The one with the big one. Yes. Okay, so this is a mortar and pestle. You guys don't have one. I looked everywhere to find like a really small, cute mortar and pestle, but you smash solids up in chemistry, okay? So what you're gonna do, you have your Alka-Seltzer, and I couldn't find a really good mortar and pestle. So you're gonna pretend, and you can use either the water bottle to crush the Alka-Seltzer, okay? We want this to be as much of a powder as possible. It doesn't have to be a completely fine powder, but you do want to crush it up as much as you can. If this isn't really working, you can try your 10 ml beaker, because we really don't. In a real lab, we would use a clean mortar and pestle, but this is not a real lab. I mean, we're not in a real laboratory. Maybe you live in a real laboratory, but probably not. We would definitely want to make sure that we had everything clean because, of course, this is gonna, could contaminate the sample. You can continue crushing. Okay, Diego's gonna continue crushing mine for me. And once you've got it crushed up, then you're going to weigh out some of the powders. So let me pull up. Okay, what did you do? What was that laugh? Oh my God. Clearly you did not get the science genes. Okay, I'm just joking. Both of your parents were chemistry majors. Okay. Let me present. All right. So, oops. There we go. Okay. In the lab link on Schoology, it gives you the instructions. And so what we are, so we did number 4. You're doing number 4 right now. And then once the powder, once you've got a decent powder, then we're gonna weigh out a portion of the Alka-Seltzer. We're going to do the same experiment 3 times, and we're gonna change the amount of vinegar and water each time. So if you have a balance, then you're gonna measure out 1 gram of Alka-Seltzer. If you don't have a balance, that's fine. You'll just divide the Alka-Seltzer into 3 parts because 1 tablet is like 3.1 grams or something like that. So you'll just divide up your Alka-Seltzer into 3 parts, and then you'll use 1 part each time. And like I said, I ordered more of the balances, and so then I will bring those to you once those come in. So what you will do before we weigh out the Alka-Seltzer is the instructions tell you to take your 25 ml beaker and then your 10 milliliter graduated cylinder. And it tells you, to measure 10 mils of distilled water. So sometimes we can use tap water, and sometimes we need actual distilled water. So you should have a bottle like this. And the little D in front of H2O means distilled water. Sometimes you need distilled deionized water, and that would have 2 little D's in it. So if you ever do research in a lab or work in a lab like in college, it's really good to try to get into a lab if you can. They would be labeled little d or little d. So this should have 30 mils in it. So the first thing you want to do is measure out 10 mils of water. Oh, whoops, mine's a 5 mil. Y'all should have a 10 mil. This is, yeah, you here.
My equipment, you might
I know. Okay, good. So you've got your powder? Okay, good. That's fine. That's fine. That's good. Okay, so measure out 10 mils of water. Yeah. And then pour it into your beaker. You don't want to use the beaker to measure the 10 milliliters because the accuracy of the beaker is low in comparison to the graduated cylinder. So it's a good habit. And don't forget that when you measure with a graduated cylinder, you want to have it at eye level, and then you get down and you measure in that way. These are plastic graduated cylinders, so you won't have the meniscus. With glass, because of the chemical composition of glass, you do get that adhesion, and you see the meniscus, and then you would measure always from the bottom of the meniscus. But these are plastic, so you don't, it's hydrophobic, the plastic. So you don't get adhesion. The water molecules are sticking together. You get cohesion, but no adhesion. All right, so that's, okay, you get that from me. That you get from me. Okay. All right, so we've got ours crushed up here. Go ahead and add your 10 mils of water to your beaker. And then on your balances, things you have to be careful. You want to turn it on, and then you'll notice that you can measure in ounces, in grams, etc. Oops, must be on the table. Oh my God, you got to be kidding me. Okay, so when you turn it on, you need to look at the units and make sure it's in grams and not in ounces. So one of the buttons will let you scroll through the units. So you want to make sure it's on the little g for grams. And that is the first thing you should always check on balances, because a lot of balances can measure in different units. Check the units. And then the second thing you need to do is called tare. So T-A-R-E. Tare is how you basically zero out the balance. So that's how you set it to 0. So you want to turn your balance on, check the units, tare it, not like tear it, but tare it, T-A-R-E it. And then when it says 0, you put your weighing paper on top and then let it settle down. And then you hit tare again. Okay? Then the balance doesn't see the weighing paper anymore. And then when you add your solid onto the weighing paper, you're only measuring that solid. Okay? But we're not going to measure the solid yet. What you're going to do is, oh, dang it.
What?
You're supposed to measure the beaker first. Okay. Don't sass me. Okay. Sorry. We need to measure the beaker first. Okay. So pour your water back into your graduated cylinder. I'm sorry. Totally my bad. Yeah. Totally my fault. What we're going to do is we're going to measure the beaker. And then you're going to write down the mass of your beaker. Then we're going to add water to the beaker. And then we're going to write down the mass of the beaker plus the water. And then we're going to measure the mass of the Alka-Seltzer. And you want 1 gram of the Alka-Seltzer. Okay. Okay, so we will turn on the balance, check the units, it's grams. Tare it, let it go to 0. Okay? Then you'll put your beaker on top of your balance. And then on a piece of paper, write down the mass of the beaker. Okay? Then we take the beaker off, and then we tare it again. You're going to tare a lot. Then the second mass that you want will be the mass of the beaker plus the water. And then we'll take the beaker of the water and put it to the side. Perfect. So always turn on, check the units, tare, then you put whatever you're measuring on, and then you get the mass, and then you take it down. So you need the mass of the beaker plus 10 mils of water. And then you want to measure out 1 gram of the Alka-Seltzer powder or chunks. Okay, it doesn't have to be a full powder. So you'll use your little scoop and you'll scoop onto the paper until you have 1 gram. So I'm going to do that real quick. I want them. Can they? I'm trying. There we go. Oh, there we go. I'm trying to make it so that they can see my hand. Okay, can y'all see my hands? Go look on your computer so you can see.
Pretty funny you just accidentally turn the computer off.
I know, that's what I'm afraid of. Okay, good. Yay. Awesome. So what we're going to do is we know the mass of the beaker and the water. We know the mass of the Alka-Seltzer. When the Alka-Seltzer reacts with the water molecules, one of the byproducts is carbon dioxide. We're going to be able to determine how many molecules of carbon dioxide is released, because when carbon dioxide is released as a gas, it will take its mass with it. And right now, the mass of the carbon dioxide is still inside the Alka-Seltzer. So we're expecting the mass of our beaker, water, and Alka-Seltzer, we're expecting that total mass to decrease. Okay? So we know the mass of the beaker, we know the mass of the water, we know the mass of the Alka-Seltzer. So you add the Alka-Seltzer to the water and it should be fizzing. So you stir it and you stir it a little bit. We want it to react completely. So you'll have to stir it a couple times. Diego?
What?
Oh, we don't have it. Go get batteries. Go get 2 of the little bitty ones.
Triple?
Yeah. I think they're in the hall closet. All right. You might need to stir it again, and you can just use one of those scoops as a stir bar. We want the reaction to go to completion. And once it stops bubbling, or after 5 minutes total, we're going to put it back on the scale. This is there we go. Oops. So turn the scale on, tare it, goes to 0. Then you put your beaker back on the scale. And if you have any little bubbles, you want to stir to let the bubbles release, because that's trapped CO2. So you write that mass down, and that'll go in your data table. That's on Schoology. You would expect the mass to go down because the CO2 has been released as a gas, so it's no longer in the Alka-Seltzer. And for the next 2 steps of the lab, you will repeat this process 3 times. The first one is with 10 mils of water. Then you will see the instructions again. You'll perform the experiment using 10 milliliters of vinegar. Then the third time, you will add 50% vinegar and 50% distilled water, so 5 mils each. And then you will add these values to your data table, okay? And then you're going to graph the data. You can look back at the introduction to make sure you have everything on your graph. And the place where you will write your answers, it allows you to upload a picture. So you will take a picture of your graph and then upload it into Schoology. And then you will write a short results and a short discussion. So in the results, you will use these terms: increases, decreases, change. And remember, the results are very just factual. And then in the discussion, you will relate the results to the hypothesis. And the hypothesis that we are trying to answer is up in the beginning, okay? We are determining how Alka-Seltzer reacts with these two different solvents, 100% water, 50% vinegar, and 100% vinegar. So vinegar is acetic acid. When you read the introduction, you'll see that Alka-Seltzer, you probably heard of it before, but you drink Alka-Seltzer because you have acid reflux, or your stomach that makes a lot of acids, of course, because it's digesting food. Sometimes you can make too much, or you don't make enough of the chemicals that would neutralize it. So Alka-Seltzer is used to neutralize acids. And you're comparing how well the Alka-Seltzer works in 3 solutions that differ in how acidic they are. So the vinegar is acetic acid. It's an acid. So we would expect the Alka-Seltzer reaction to be stronger in the acetic acid. So we would expect more CO2 to be produced when Alka-Seltzer reacts with an acid. It will go, that reaction should go to completion more times. That reaction should occur faster, and there should be more reactions. So the rate of reaction should increase in the vinegar. So you're going to expect to see the biggest change in mass with the 100% vinegar. And then the middle change in mass would be the 50%. And then the smallest change in mass you would expect with the water. So those would be the expected results. If that's not what you get, that's completely fine. You always give your results. And when you read over the lab and the pre-lab, it says everything that I've just told you. Under normal circumstances, you would have had access to the pre-lab and the lab. In the future, you'll have access to all the labs and the pre-labs ahead of time. So I also have some reminder videos in here if you want to watch them. Any questions from anyone? You'll use the same beaker all 3 times. So you'll do the experiment, collect your data, then you can just pour this into a sink. And then you'll just want to rinse it with water and then dry it out with a paper towel. And then you'll do the same thing again, except you'll use 100% vinegar during one trial, and then you'll use 50% vinegar on the other trial. Okay, let me check again. Oh, see, yes, I have a stir bar. Y'all don't have a stir bar. This is a glass stir bar. So you guys can use either the red, the yellow, or the white scoops. Whichever one you used to measure, well, any of those 3 you can use. After you're finished with each experiment, you'll always rinse with just tap water. Don't add any soap. Just rinse with water and then dry it out with a paper towel, and that should be fine. With soap, we're going to try not to use it just because the soap can actually leave a residue and then change our results. Unless we work with something that's not water soluble, we'll just rinse with warm water and dry it. What should we put in the dip if you don't have a balance? Okay, so if you don't have a balance, then I'm going to email you your data. So I've already done the experiment. I have my results. So put your name in the chat if you don't have a balance, and then I'll email you the data. Okay, let me write this down. So Maggie. Erica. Mary Catherine. And then Ethan. All right. Anybody have any more questions? Okay, so I will stay here for the next 20 minutes. If you want to stay and finish the experiment so that you can ask me questions, you can do that. If you want to just go offline and finish the experiments, completely fine. I will email you guys probably tomorrow, and we can start the discussion on the class structure. If we want to keep it 3 hours twice a week, or if we want to split it up. I've made a couple different models, but we can go ahead and talk about that. I can stay online and talk about this all day long, but I know that being at home and online for 3 hours may be a beating. And then if we were at the school, I would be giving you all kinds of chocolate and candy. And that makes the 3 hours a lot more enjoyable. And I'm a lot funnier in person than I am online. So it would be more entertaining. So we'll just decide as a group what we want to do. If you have questions, email me. If you have questions, please text me. We are moving quickly. Your parents are paying for the attention that I will give you. So I am here for you. I don't mind if you text me and ask a million questions because we're going really fast. That's to be expected. All right. Okay, then I'll stay here till 3:30. You're welcome to stay, but if you want to log off and just finish these, not a problem. All right. And then I'll see you guys on Thursday at 12:30. All right.